Good day. So our topic is all about detection of Entamoeba histolytica using Colloq detection and watershed transform. So for the background, Entamoeba histolytica is a pathogen that causes amoebiasis to a patient. We also discuss in the paper the process called fecalysis, in which series of tests will be done to the stool in which observing pathogens is also done. So we also discussed the following techniques that is used in our system, which is the Colloq detection, an algorithm used to detect edges in an image, and a watershed transform, also an algorithm used to determine circles that overlaps. So the main objective of the study is to create a device using Raspberry Pi that will help medical technicians to examine the stools and use Colloq detection and watershed transform in determining the Entamoeba histolytica. And for our significance, the main contribution of this study is to make the detecting process automated through image processing, and this will also benefit medical technologists on their laboratory exam in comparing their results to the device result. So shown on the figure is our conceptual framework, which consists of input, the processes done, and the output. So we have the input, stool sample, and the following are the processes to convert the image to grayscale, apply cutting edge, apply watershed, and apply Howe transform. And for our output, the size of the stool, the presence of the chromatoidal body, the number of nucleus, and the analyzation if the stool is an Entamoeba histolytica or not. Shown in the photo is the proper setup of our prototype, which consists of endoscopic camera placed above the microscope, the monitor for display, the mouse for navigation, and the Raspberry Pi for the system itself. Next is the sample graphical user interface of our system. So we have here the number of nucleus, the chromatoidal body, and the size of the cyst, which is our primary objective to be detected, because through these three parameters we can tell if the stool is positive on Entamoeba histolytica. And of course, the graphical user interface which displays a positive result. So we have here the number of nucleus, which is 3, the presence of the chromatoidal body, and the size of the cyst. So for our data presentation, we gathered 10 samples and used the following tables. First is the cyst size comparison table. It consists of the sample, the size of cyst for manual observation, which is done by the medical technologist, the size of cyst for automated observation, which is done by our system. Next is the number of nuclei comparison table that consists of, again, the sample, the number of nuclei for manual counting, again done by the medical technologist, and the number of nuclei for automated counting, again done by our system. So here in table number 2, as you can see, results for samples 2 and 3 shows 5 nucleus detected. With that, these two samples should be declared negative, since detecting Entamoeba histolytica should satisfy 3 results at the same time. If one parameter came out as negative, the pathogen will be declared negative from Entamoeba histolytica. And for our last parameter, which is the presence of karyosome comparison table, which shows if the karyosome, or what we call the chromatoidal body, is present within the cell. So our system will tell if the stool is positive on Entamoeba histolytica if it satisfies the conditions stated earlier. First is a size condition. The size should only be 12 to 15 micrometers. And next is the number of nuclei. Entamoeba histolytica should contain 3 or 4 nuclei only, and the presence of the karyosome. So for our testing, shown is the proper setup of the system. So the endoscopic camera is attached to the microscope, and the monitor will serve as a display. Next is to find the bacteria around the specimen. Once a bacteria is found, the process button shown in the graphical user interface will be pressed. Next, After the image taken is processed, the system will show the result together with the following parameters: the number of nuclei, the presence of the chromatoidal body, and the size. Also, the processed image will also be shown. So for our last table, we have our confusion matrix. To tell if it is a true positive, our system should detect positive and the medtech should also detect positive. To tell if it is a true negative, our system should detect negative and the medtech should also detect negative. Lastly, to tell if it is false negative, our system should detect negative while the medtech should detect positive. Out of 10 samples, 8 came out as true positive, 1 came out as true negative, and lastly, as shown, 1 came out as false positive. So for our conclusion, the study was able to develop a system that could identify a bacterium named Entamoeba histolytica using image processing techniques. The samples were successfully taken using the endoscopic camera attached to the microscope. The techniques used by the system were can edge detection, watershed transform, and Hou transform. The parameters needed to determine if it is a positive result are the number of nuclei present, the presence of karyosome, and its size. Thank you.